In the last sessions we have discussed about the basics of renal physiology. Today I will be discussing with you regarding the glomerular filtration and how the filtration barrier is maintained. The kidney gets approximately 20 to 25% of the cardiac output, and every day around 180 liters of fluid is filtrated from the glomerulus. So let us look into the structure of the glomerulus. So the glomerulus has got an afferent arteriole, the artery that is bringing blood to the glomerulus, which divides into network of capillaries, and finally the blood leaves the glomerulus via the efferent arteriole. So this is the efferent arteriole and this is the afferent arteriole. The afferent arteriole is thicker than the efferent arteriole and it is under control by neural control and also by the macula densa and the juxtaglomerular cells, about which we will discuss later. Here is a situation where an arteriole leads to capillaries, and from there again an arteriole is formed, and the pressure inside the glomerular capillary tuft is very high compared to other capillaries, and this is the major driving force for the fluid to enter the Bowman space. So this is the filtration pressure that produces an ultrafiltrate, and this is going in the Bowman space. The Bowman space is shown here. It has got two layers. One is the visceral layer. So this is the visceral layer of Bowman's capsule, and it is continuous with the parietal layer, and that form later continues as the proximal convoluted tubule. So around 180 liters of fluid is drained or filtered every day, and that is reabsorbed from various parts of the tubules. Hope this point is clear to you. And the visceral layer of Bowman's capsule, these cells are modified and they form what is called the podocytes. The podocytes are the modified visceral epithelial cells of the Bowman's capsule. It is called podocyte because it has got food processes that is wrapping the glomerular capillaries. So this is the podocyte, and this contains the food process that is wrapping around the glomerular capillaries. Now we all know that the ultrafiltrate doesn't contain protein or cells, and we will be seeing a clinical case where the urine shows proteins 3.5 grams and it has got RBCs. This means that from the glomerulus, protein and RBCs are leaking into the Bowman space. Normally this is not happening, and the urine here shows, the heat test shows a cloudy urine, meaning that there is protein is present, and the patient has got puffiness of face, especially periorbital edema. You can see around the eyes there is puffiness. This is a feature of renal disease. The massive proteinuria that occurs is classical of nephrotic syndrome. So in the following session we will be discussing about how there is a barrier for the excretion or the barrier for the transfer of cells and proteins into the urine. So we have learned about the glomeruli. So the glomeruli has got basically there is an afferent arteriole and there is which forms the capillaries, and then again it forms the efferent arteriole, and the efferent arteriole again branches, and it branching around the tubules, that is called the peritubular capillaries. Some of them descend into the renal medulla, and they are called the vasa recta. So from this vasa recta and peritubular capillaries, venules are formed, and the blood is leaving the kidney via the renal veins. This part has been discussed in the previous session. So this picture shows the electromicrograph of the glomerulus of a mouse, and this is the glomerulus, and in the enlarged view here you can see that the glomerular capillaries are wrapped by the food processes of the podocytes. So this is a bit little enlarged view. Here you can see that these are the podocytes with the food processes which are wrapping all around the capillaries. Hope this point is clear to you. And this is a part of this is taken, and here it shows this is the inside of the endothelium, which shows the blood. This is the capillary endothelial cells. This is the basement membrane, and these are the modified visceral epithelial cells, otherwise called the podocytes. So we will go a little more into the detail. So this is the capillary. So we know that the afferent arteriole enters. This forms the capillary network, and it leaves us the efferent arteriole, and afferent arteriole is bigger than the efferent arteriole. So here this is the inside of the capillary, this is the inside of the capillary, and these are the endothelial cells. So lining these are the endothelial cells, and the endothelial cells, these are sitting on the glomerular basement membrane. This is the glomerular basement membrane, and towards the outer side, that is inside the Bowman's space, here are the podocytes. With the food process of the podocytes, they are wrapping around the capillary, and the basement membrane is formed and nourished by or maintained by the podocytes. Any dysfunction of these podocytes or the food processes can lead on to disruption of the structure of the glomerular basement membrane, and you should try to draw this picture and learn it. And here again we are showing this is the capillary endothelial cells, and one of the important point of the capillary endothelial cells, suppose this is the take as the capillary endothelial cells, this has got fenestrations. So the capillary endothelial cells has got multiple holes. These are nothing but channels, protein channels, and these allow molecules which are larger than 7 or up to 70 to 100 nanometers. So the pore size is like this. Materials which are larger than cannot pass through. That is why the RBCs and also the neutrophils cannot pass through, and normal urine doesn't contain this. So what can pass through? Amino acids can pass, glucose can pass, electrolytes can pass, and small quantities of smaller size of proteins can also pass. In addition to the pore size, the charges of the membrane is also important. The capillary endothelial cell has got a glycocalyx over it. This is negatively charged, and that is why albumin, even though the size is smaller, the albumin cannot pass through. The negative charge repels the things. So not only the pore size but also The charges are also important. And this is sitting on this is the basement membrane. The basement membrane has got a thick inside, a dense part, and that is called lamina densa. The lamina densa is formed of collagen, type 4 collagen. So it is the very important thing to remember. It is formed of the type 4 collagen and other proteins like laminin. Systemic diseases affecting formation of type 4 collagen can lead on to abnormalities in the glomerular basement membrane. Now the next structure is these are the podocytes with the food processes wrapping the capillary network, and in between there are spaces, and these are called the filtration slit. So this is the filtration slit, and the filtration slit is again, there are proteins, mainly the major protein is the nephrin, that is forming barriers there, and that produces what is called the slit diaphragm. So the slit diaphragm produces reduction again the size of the filtration slit. So the filtration slit is guarded by the proteins. It will act something like a zip. Nephrin, podocin, different types of proteins are there. Abnormalities in this protein can also lead on to disruption of the filtration barrier. So basically there are three structures are there as the filtration barrier. This filtration barrier is constituted by one, the endothelium, second is by the basement membrane, and third is the podocyte. In addition to the charges, they are negatively charged. The endothelium has got a glycocalyx which is negatively charged, and also the basement membrane. We'll go a little more into detail of the basement membrane. So here this shows this is the endothelial cell. So here you can see this is the capillary endothelial cell which has got fenestration. So fenestrated, that is a characteristic feature, the fenestrations. And then it is sitting on the basement membrane. The basement membrane has got three parts. This is what I have told about as the lamina densa. This is formed by the collagen tissue. It is actually a helix, triple helix of proteins. And on the inner aspect, towards the endothelial side, it is called lamina rara interna. Rara means it is less dense. And the outer side, towards the podocyte, is called the lamina rara externa. So there are three parts for the basement membrane. Sometimes antibodies can be formed against the basement membrane, the proteins of the basement membrane, and that is called these antibodies are called anti-glomerular basement membrane antibodies, anti-GBM antibodies that can produce, they can destroy the basement membrane and can lead on to massive proteinuria, or it can lead on to passage of RBCs through the urine. The same way the podocyte, I have said, the podocytes are producing and maintaining the basement membrane. So disruption of the podocyte or the food processes can lead on to disruption of the filtration barrier. So this is the glomerulus, and you can see clearly how these three layers are arranged. Now, few words regarding the filtration slit. The space between these are the podocytes. You can see that this is the podocyte. So if we should draw the podocyte, these are the food process, the podocytes. The space between the podocytes is bridged by proteins. The major protein is nephrin. And this is the filtration diaphragm. and this nephrin is in connection with the cytoskeleton of the podocyte, and mutations in the nephrin gene can lead on to abnormalities in the filtration barrier and can lead on to proteinuria. So we go to the clinical aspects. So far we are discussing about the structure of the filtration barrier. It includes fenestrated endothelium, the three-layered basal membrane, the foot process of the podocytes, and we also mentioned about the filtration slit and the filtration diaphragm, which is produced by the nephrins. Now we go to few things discuss regarding the clinical importance. First and foremost, we have already said the collagen, type 4 collagen is an important constituent of the glomerular basement membrane. Some congenital diseases, typical the classical condition is Alport syndrome, where type 4 collagen is abnormal. This can lead on to proteinuria, hematuria, because the filtration barrier is lost. And it can also produce abnormalities in other parts, like it can produce sensory neural deafness. This is an inherited condition. Second condition is that the basement membrane. We have mentioned about the basement membrane. This is the fenestrated endothelium, which is sitting on the basement membrane, and here comes the podocytes. The basement membrane is unusually thin. This is called a thin basement membrane disease, and this again leads on to hematuria because the RBCs can pass through. Another important condition you should remember is the nephrotic syndrome. Nephrotic syndrome is characterized by massive proteinuria. More than 3.5 grams per day is usually the criteria taken for the massive proteinuria of nephrotic syndrome. And again, this is due to the abnormalities in the filtration barrier. And the patient usually presents with generalized edema because of the hyperproteinemia that produces changes in the Starling forces, and other abnormalities we will study at a later time. And one of the important conditions which is very common in children among the nephrotic syndrome is the minimal change disease. Minimal change disease. The term itself means minimal change means on histopathological examination there are not much changes. It is effacement of the food process, that is food processes are lost. So we have already mentioned that the podocytes are concerned with the maintenance of the basement membrane, and abnormalities can lead on to disruption in the filtration barrier that leads on to proteinuria. So another common condition that produces nephrotic syndrome is diabetes mellitus. In diabetes mellitus, what is happening? So there is what is called a glomerular sclerosis is occurring. Outside the capillary, there are cells called mesangial cells, which secretes materials together forming what is called the mesangium. In diabetes, there is the nodular sclerosis occurring that will be destroying the glomerulus and produces what is called the KW syndrome, otherwise called Kiemenstein-Wilson lesion, that produces nephrotic syndrome. In Goodpasture syndrome, the antibodies against the basement membrane also damages the basement membrane of the lungs and leading on to hemoptysis. The next session, we'll be discussing about the regulation of the renal blood flow. Hope this was useful to you. Thank you.